Hi, welcome back everyone. Let's continue our discussion on fitness landscapes. So far we have seen two concepts. One is the concept of sequence space, and the second one is a concept of Hamming distance. So from the context of sequence space, we saw that for any sequence of length L, the number of possibilities of DNA sequence, unique DNA sequence, is 4 to the power L, and Hamming distance is defined as Hij, which is the distance between sequences I and J, and is simply equal to number of nucleotide positions where sequence I and J are unequal, are not the same. So we saw that if sequence I is AA and if sequence J is AT, then Hamming distance between these two sequences is simply equal to 1, because at the first position they are identical, at the second position they are not identical. So this is not true. So the number of positions where they are not the same is 1, and that is the value of Hamming distance Ij. So having made these nodes, we represented each sequence by these nodes. The next step we will do is that we will connect nodes I and J if Hamming distance Ij is exactly equal to 1. So in the DNA sequence of length 2, what we have is 16 nodes. What we need to identify is here is every pair which is one Hamming distance away from each other, and should that be the case, then we join the nodes with an edge. So let's do this, but let's first make these nodes. Let's make these nodes. Okay. So what you should see is that A and AT, the Hamming distance between these two nodes is 1, because they are identical at the first position A, but not identical to each other at the second position T. So these two have to be joined with a edge. Same with AA and AG, because identical at first position, not identical at second position. So these two have to be joined also. And the same logic applies to AC. Let's continue and see and find other partners for this node AA. AA will also be connected to TA, because these two are identical at the second node, but not identical at the first node. So these two have to be joined with each other also. AA will also be connected to GA because of the same reason, identical at second position, non-identical at first position. So these two are joined also, and similarly AA will be connected to CA. And you will see that AA should not be connected with the remaining 9 nodes, because for each one of these 9 nodes, the Hamming distance between AA and them is 2, and our rule is that we will only connect 2 nodes if the Hamming distance between them is is 1. So, the number of partners of each node in this case is simply equal to 6. By the same token, we can see the number of partners of another node. AA will be partner, but that has already been connected. AT will be partner with AG and AT will also be partner with AC. Similarly, AT will be partner with TT and with GT and with CT. So, again the number of partners for AT as a node will also be 6. So, every node will have 6 partners, and the way we get that is because length is equal to 2. So, if I have any sequence, let us say AA, and I am looking at Hamming distance 1 partners, I can only mutate one position. I can't mutate both the positions, because if I mutate both positions, then Hamming distance becomes 2. So, for Hamming distance 1, I can only mutate one of the 2 positions. Now, I could mutate the first position, but I could mutate it in 3 different ways. I could change this A to G, T or C. So, that gives me 3 nodes which are neighbors of AA by only Hamming distance 1. Similarly, I could mutate the second node also in 3 different ways. I could mutate it to G, T and C, which is 3 nodes. So, 3 plus 3 is equal to 6 nodes. And we can generalize this, that if we have a sequence of length L, then the number of nodes in the sequence space is equal to 4 to the power L, that we have already seen. But in this map that we have, how many partners will each node have? And it is easy to figure that out, because I could, when I am looking for partners, each of which is at a Hamming distance 1, that Hamming distance 1 could be because of dissimilarity at position number 1, which could happen in 3 different ways, or that Hamming distance could have Hamming distance could be 1 because of dissimilarity at position number 2, which could be in 3 different ways, and so on and so forth. So, every position offers me a chance to make a mutation in 3 different ways, and hence, and since I am only allowed one mutation, the total number of neighboring nodes, which are connected nodes, is going to be 3 times L. 3 options for every one of the L positions that this sequence has. And this is the reason why in the previous case we got the answer 3 into 2, which was 6 neighbors for every position. So, that gives me some idea of what to connect and not to, and I will end up with a structure which looks like this. Here we have only drawn completely for these 2 nodes AA and AT, but the entire network is going to be much more messier than this. So, what do we do next? Now, there is only one thing left, that in this diagram we are going to have to draw that diagram again. Node number 1, node number 2, going all the way to node number 4 to the power L. We are going to do a hypothetical experiment, and our hypothetical experiment is as follows, that for a sequence of length L, I have 4 to the power L options, unique sequences. I will list them all here. So, let us say this is sequence number 1, which is represented by node 1, this is sequence 2, sequence 3, sequence I, sequence j, going all the way to sequence 4 to the power L. I will do a thought experiment now. The thought experiment is, imagine that I have E. coli, but it does not have DNA, and obviously that is not going to be possible, but this is a thought experiment. In this E. coli, I will add DNA which is corresponding to, it is corresponding to sequence which is represented by node 1. So, let us say this is the sequence space map and connections are made where L is 5 into 10 power 6, which is E. coli genome. So, each one of these sequences is of length 5 into 10 power 6, and in my thought experiment I take E. coli's cell membrane and everything that comes except for its DNA. In place of DNA, I will take the sequence associated by node 1 and put it inside this E. coli and add this to a flask where I have growth media, and in this growth media I will add this E. coli whose DNA is represented by the DNA sequence corresponding to node 1 in the sequence space map, and I will measure the growth rate here, which will be a proxy for fitness. So, I will do this experiment and I will record against node 1 the fitness associated with the bacteria that was carrying the DNA sequence represented by that particular node. In my thought experiment, I will do this again, but this time the DNA that will be carried by E. coli will be the DNA sequence represented by node 2, and hence I will get the fitness measure associated with the sequence associated with node 2, and I will write this as F2. And I will keep on doing this till I am done with all of these sequences of 4 to the power L. So, eventually giving me F 4 to the power L. And we say that this is a thought experiment because of the sheer numbers involved in this. If L is 5 into 10 power 6, there is no way we can do this experiment for 4 to the power 5 into 10 power 6. But anyway, in this thought experiment, E. coli carrying DNA represented by nodes is added to growth media and the growth rate is measured. So, we have a number corresponding to every one of these nodes that we have drawn here. And in the final step to get to fitness landscape, what we do, so now we have to imagine this sequence space as nodes in this plane. So, let us do this on the next slide. So, I have this sequence space nodes in this particular plane. Going all the way to 4 to the power L, and let us say 1 and 2 are connected, 3 and 4 are connected, I and j are connected, and so on and so forth. What these connections mean is that these two sequences are separated from each other by only one Hamming distance. And now remember, corresponding to sequence 1, I have a fitness proxy growth rate which is F1. Corresponding to sequence 2, I have fitness proxy corresponding to F2, and so on and so forth. Going all the way to 4 to the power L, I have a fitness proxy which is F4 to the power L. So, now what I am going to do is that this landscape exists in this flat plane of the screen. I will go to a particular node which is node 1 now. I will see what is the fitness value F1, and I will lift this node up from the screen by a distance which is proportional to its fitness value, which is F1. So, let us say that node 1 is its fitness units, let us write some numbers here. Let us say F1 was simply equal to 1 and F2 was simply equal to 2. So, what I will do is I am adding another axis to this screen which is perpendicular to the screen, and now I will go to node 1 and lift it perpendicular to the screen by a distance which is equal to 1 unit. I will similarly go to node 2 and lift it perpendicular to the screen, but by a distance which is twice as much as how much I lifted the node 1, and so on and so forth. I will do this for every node in this sequence space. Again, so this is akin to me adding another axis which is perpendicular to the screen and then going to each node and lifting it by a distance which is equal to the fitness of the organism which is carrying that particular DNA sequence. If I do this, I hope what you are able to visualize is that we will get a fitness landscape type of a structure which will be ups and downs. There will be many ups and downs associated with this. The nodes which correspond to sequences which exhibit very high fitness will be the peaks. Some nodes will have fitness which is equal to 0, they will remain on the screen, they will be the troughs, and so on and so forth. And this 3-dimensional structure is called fitness landscape, where the plane is occupied by all the sequences and the perpendicular axis is the fitness corresponding to each one of these sequences that we are talking about. I hope that idea of fitness landscape makes sense. So, viewed from top, fitness landscape could look like this. So, if these two axes are, this is the plane of sequence spaces. What you should be viewing this is like a map reading exercise where these regions represent heights. So, this is the highest peak, which means this sequence represents the sequence corresponding to this particular node is the fittest one, and as I move away from this, I am moving towards lower fitness values. Let's also look at it another possible way. Let's say that we have these sequences on one axis only, AA, A, we rewrite this, AA, AT, AG and AC, and let us say this is fitness. Now, imagine that this node was this particular fitness, AT was slightly higher, AG was significantly higher, and AC was really bad, really low fitness or really bad genotype. Now, so the structure we get is because AT and AA and AT are separated by Hamming distance 1, these are all connected to each other. Because the distance between them is exactly 1. And this is the 3-dimensional structure that we have now. If there is a population at AT, so let us imagine a population of bacteria which is present at node AT. One member of this population could acquire a mutation and come to AC. This movement from there to there leads to decrease in fitness. The transition that happened here was AT going to AC. So, the mutation was T going to C. This mutation has led to decrease in fitness, and hence this mutation is a deleterious mutation. As a result, when one of the offsprings of these individuals lands up here, natural selection is going to remove this. Natural selection weeds out deleterious variants. And the same story is going to happen if one of the individuals acquires this mutation such that a offspring with genotype AA is born. In that case also, this leads to decrease in fitness. This is also a deleterious mutation, and hence natural selection will get rid of the AA individual from the population. However, there will eventually one of the individuals is going to land up at this particular genotype. Because it is Hamming distance 1, this mutation, a beneficial mutation will take place such that an individual with genotype AG is born. So, from AT the population moves to AG, and now because this has resulted in increase in fitness, this is obviously a beneficial mutation. And natural selection selects this particular variant, and as a result of this, the progeny of this individual are going to outcompete the progeny of the ancestral population, which will be outcompeted by AG genotype, and hence the entire population, given sufficient time, the entire population transitions from being at AT to being at AG. That transition takes place. What is important to recognize here is that in this view of evolution of populations, any variations that are of lower fitness, that are variations that are obtained via acquisition of deleterious mutations, are movement down on this mountain structure that we have made, and natural selection gets rid of them, and only variations which lead to an upward movement on this landscape are beneficial mutations and hence are selected by natural selection. As a result, this adaptive trajectory of the population is always going to be in an upward direction towards increase in fitness. And this is the metaphor of fitness landscape and the geometric interpretation of fitness landscape that is used to visualize evolutionary processes in this context. So, next video onwards, we are going to look at actual concrete examples from last few years. We will discuss 3 specific examples from the last few years, which have experimentally tested these ideas and study how populations move on these landscapes. And we will continue that in the next lecture.